Hey everyone, how is everyone? My name is Artin and I am a first year medical student, and I'm gonna give you a brief crash course on endocrinology and kind of delving into one of the axes of endocrinology. Okay, so what is endocrinology? This is a little funny picture, and it's a little schematic. The farmer is saying, the cow said, you might know what happened to the crate of animal growth hormone I had stashed in the barn. I thought it was kind of a cute little picture about endocrinology and hormones, the study of hormones. Okay, so briefly, what are we going to cover today? I'm going to give you, like I said before, a brief introduction, some general characteristics about the endocrine system. We're going to cover the HPA axis, and then we're going to open it up to any questions and any concerns you may have. All right, so what is endocrinology? So endocrinology has two major functions, okay? One of the functions serves the developmental portion, and the other one is in the functioning organism of how endocrinology works. So in the developmental portion of endocrinology, you have what we call here young Harry Potter, and I don't know what happened, he became this person, and I forgot the movie, I think it's Pegasus or something, I don't know. But this kind of describes the developmental portion of endocrinology, like puberty, and represents coordinated transformation of cells. So, and that's with various hormones, which we'll get into in a bit as well. So it transformed or metamorphosized young Harry Potter to this, okay? The second portion of endocrinology is within the functioning organism, and that's when we have hormones responding to stress or responding to any environmental trigger. Over here you have stress, you have Harry Potter with Hermione kind of chasing over a Dementor probably, I don't know. And over here you have, like, an example would be sexual behavior, arousal. Arousal could very much well be a part of this as well, okay? And it's a coordination of cells in response to your environment or in response to any stimuli. So what you need to know, I want you guys to know the differences between lipid-soluble and water-soluble hormone systems, and what normally determines hormone activity and the consequences of receptor downregulation, what is meant by permissive action, and properties of hormone receptors and signal transduction. Okay? So, signal transduction. We have two sets of hormones. You have steroid hormones, and you also have what we call peptide or protein hormones. Does anyone know what steroid hormones are? No, okay. So that's fine. So steroid hormones, the way you could think about it, in the stem of the word, you have oid, okay? Oid means, I guess, similar to. So you have steroid, cholesteroloid. So it's like a similar thing to cholesterol. That makes sense because steroid hormones are actually derivatives of the molecule cholesterol. So if we all know what cholesterols are, we know that they're what? Are they lipid soluble or are they water soluble, guys? Yes, they are lipid soluble. So that's one unique feature of steroid hormones. One other thing we have to know about steroid hormones is that they are protein bound. Okay, so steroids are not freely floating in your blood; they are actually attached to proteins. These are called binding globulins. We're going to learn about that in a bit. So steroid hormones are also very slow. They're very slow acting, slow everything. Anything about steroids, are you going to think about it being very slow? So since they are slow, they also have particularly long half-lives, and that's really important when you're trying to measure any blood levels of a hormone or steroid hormone. And so they also need time to potentiate their action on their target tissues or their target organs. Now that's also in contrast to peptide hormones. Peptide hormones are very fast. Think of a peptide, pep, pop. They're very fast. They're fast acting. And since they are fast acting, they also have very short half-lives, okay? It's the opposite of steroid hormones. And another unique feature of peptide hormones is that they are not bound to any protein carriers, and they are freely floating in your blood. Okay, that's one unique thing about peptide hormones. And since they are not bound by proteins, they easily go to your kidney, and your kidney excretes them. So they are freely excreted, so you probably don't have many peptide hormones found in the blood. So that's one unique thing. So peptide, pop, very fast. Steroid hormones, steroid, slow, very slow hormones. So one unique thing about steroid hormones also in the signal transduction pathway is you see over here they have no receptors. Okay, so steroid hormones, since it's lipid soluble, it's able to go inside your cell. And it's able to bind to a nuclear receptor or a receptor that's inside of the cell. and that's where it could then transcribe or translate and make proteins. Okay, so that's a unique thing about steroid hormones, is that they don't have a receptor. The receptor is located inside your cell. Okay, now over here, the unique thing about peptide hormones is that they do have a membrane receptor, and this is really important. So you need to have this membrane receptor, and that's coupled with what we call a G protein. Now we learned in biochemistry, I think Dr. Walker, he mentioned about G proteins, right? So G proteins are then what catalyzes, I guess, the secondary messenger system. Okay, and that's really important. So the secondary messengers include cyclic AMP, calcium, IP3, and DAG. IP3 is what, you guys? Inositol triphosphate. We also have DAG, which is diacylglycerol. So what these do is that they phosphorylate or dephosphorylate proteins. Now what's important here, guys, look over here, this protein is already inside the cell. Okay, so this protein was not made and then bought. So the peptide hormone did not make this protein. The protein was already there. That's in contrast to steroid hormones, where they're actually able to make protein. Okay, so that's one big importance as well. All right, it's kind of weird because peptide, you think of protein, but just know that the protein is already there. And then it goes ahead and phosphorylates or dephosphorylates it. So that's also a very main portion of peptide hormone. Here's a little summary. You guys could look at this on your own, but it kind of talks about a good little compare and contrast between the two, which is an objective that I'd like you guys to know. So this is right here for you guys to have. Now I'm gonna delve into what I mentioned earlier about steroids and that they're binding globulins. It's made in the liver, and you could have cortisol binding globulin, thyroid binding globulin, and or sex hormone binding globulin. And this is important because it keeps the hormone inside the body, and it requires too much energy to keep making new hormones. So when you're so steroids are very energy intensive. So once you make them, you want to hold on to them. So you make these binding globulins, and that helps with that. Okay. So in equilibrium, so you have most of the lipid soluble hormones are bound to what, you guys? Yes, they're bound to binding globulins. Perfect. So some are also unbound, though. Okay, that's also important to note. And this unbound portion of these hormones, they're the ones who are available to the tissues, and they're the ones who actually could potentiate any sort of action. And that kind of creates a little negative feedback loop. So the total hormone for, like, let's just say cortisol over here is what's bound to the protein and what's freely bound, that's freely unbound in the blood. So that's a free hormone. That's a total hormone level. Okay. So role of the liver. So liver, like we said, is what makes the binding globulins. They also are related to modulation. So what happens if you have liver problems? You're unable to make these binding globulins. And what does that do? That's not good, because like we said before, we need these binding globulins for them to bind to the steroids. So what this actually does is then, since you don't have any, since you're going to have more free hormones, okay, because you're not having enough globulins, you actually create a negative feedback. So it's not negative. You have no feedback control anymore. So what you're going to do is you're going to have increased circulating binding globulins. And I have a little few questions over here, and you guys should be able to answer that as well. Okay. So hormone receptors, I just want you guys to know that the specificity. So hormone receptors will affect specific parts of your body, and they each have their own receptor, and the activity. And we also have to know that the receptors are never saturated, meaning that receptor availability is never the limiting factor. It's usually the amount of hormone. Okay, I have a little checking in for you guys. So disease of which of the following organs would be associated with the total low steroid hormone level? Monica?
D.
Yes, you're correct. It's disease of the kidney. Okay. And another one, 52-year-old males brought to your clinic, and they have what's important thing here is nephritic syndrome, which is problem in your liver, or problem with your kidneys, actually, I'm sorry. So which of the following blood levels will be low because of this?
C.
Perfect, exactly. Testosterone deals with your kidney, the adrenals, and that's the whole point about it being localized to your adrenal cortex with the receptor specificity. So now the HPA axis, so that's the site of synthesis, storage, and release of neurohormones, and we're going to talk about this axis very briefly, okay? So all the hormones in this axis are water soluble, and they're synthesized in neuron cell bodies, okay? And the pituitary is located in the sella tunica, which remember from physio is the bony surface of the brain, okay? So over here we have the little cell bodies. Sorry. All right. So over here you have the cell bodies over here, and they're the ones who kind of make the releasing hormones. Okay, and notice that they're all releasing hormones, which actually then stimulate for you to make more hormones, except for PIF or dopamine. PIF is prolactin inhibiting hormone. That's the only hormone that is going to inhibit or decrease the amount of total hormone in the very end. That's very important. That's a very important board question. If you sever the stalk, what's going to happen to these hormones? So over here, what's also really important is the pulsatile control. This pulsatile control means that it's not a steady infusion of GnRH or any sort of hormone. It's pulsatile, and that's very important for downregulation. Okay, that's also very important. And then here's a little summary again. We mentioned about the releasing hormones. Remember, the hypothalamus has the cell bodies, and they have the releasing hormones, with the exception of what? Prolactin or dopamine, exactly. That's a negative inhibitory effect to your prolactin. And these hormones then are then stored in your pituitary, and then whenever you need them, they're released. Okay, so here's a clinical correlate. You have hyperprolactinemia, and some causes of this, it's all in your books, but the pathology is what if you have like a pituitary adenoma, or some medications cause this as well. So it's a dopamine antagonist. So you have all these little pathologies happening, but what would be an appropriate treatment for hyperprolactinemia if it's a dopamine antagonist? You just prescribe them a what? A dopamine agonist. Perfect. All right, now this is one checking in to see if you guys are awake. Monica, what's the answer to this?
C.
Prolactin, exactly. And why is that? Prolactin is the only one that's an inhibiting hormone. So if you sever the pituitary stalk, you don't have the inhibition, you're going to increase the prolactin. All right, here are some take-home points I want you guys to know, and that is it. Do you guys have any questions? Thank you.